Hi everyone, I'm Chunhui Cai from Lishan Biotechnology. Many thanks to the Cell Regeneration's invitation to give us this great opportunity for sharing our recently publication. Here I will introduce our paper about IFN-β in glioma stem cells to all of you. First, I would like to give you a brief introduction about malignant glioma. Malignant glioma is highly aggressive and represents the most common primary brain tumors. The overall survival period is typically less than 15 months, even with regular therapy, including the surgical resection and chemoradiation. Tumor recurrence always happen. 1 year after diagnosis, about 70% of patients may facing the disease again. The 5-year overall survival of GBM patients is less than 10%, with a median survival of only 14 to 16 months. Recently, more and more researchers believe that the high recurrence of GBM was due to the glioma stem cell. And for the glioma stem cells, we all know that glioma had a complex tumor microenvironment, which includes glioma stem cells, mature neural cells such as oligodendrocytes, astrocytes, microglia, and the ependymal cells, and of course many immune cells. Although several groups isolate and characterize stem-like cancer cells in glioma and try to find the genes which regulate glioma stem cells maintenance and the tumor progression, the underlying mechanisms remains unclear. So, our group would like to deeply understand the molecular mechanisms of how glioma stem cells function. Here is our previous work in 2017. We developed a highly efficient culture system to gain human neural stem cells. Our primary cell lines show significant stemness and the ability to differentiate it into astrocyte and neuron. In order to study the differences between human neural stem cells and human glioma stem cells, we also developed a culture system to culture the glioma stem cells. Using this method, we would establish a glioma stem cell line from primary tissue in only 3 days. And these stem cells can be maintained for more than 10 passages. In this case, we could collect enough biological materials for further molecular mechanism study. Well, in our publication, we focus on the functional role of IFN-β, which is a member of interferon factors. Human IFNs induce the JAK and STAT cascade by binding to the IFN alpha or beta receptors. IFN-β signaling has been proved to inhibit cell proliferation in many types of cancers. Dick and his group tested the different IFN factors in 5 human glioma cell lines, and from their results, we noticed that IFN-β seems to have better anti-tumor effect than the others. Later, more studies tried to identify the downstream molecular mechanisms of how IFN-β functions. Here are some results. It could either inhibit miR-21 or upregulate the suppressor of cytokine cyclin 6 and inhibit JAK1 step 2 to decrease the tumor cell growth. While people noticed that IFN-β could efficiently block tumor cell growth, they started to combine it together with some chemotherapy drugs such as TMZ. As we can see here, many different glioma cell lines were invoked for the combination drug test. Most of them results confirmed that combination of IFN-β and TMZ chemotherapy increased tumor sensitivity to TMZ. However, the underneath molecular mechanism is still not clear. Except studies in glioma cell lines, several clinical reports also prove the functional roles of IFN-β together with TMZ treatments. Well, among all the publications, Ito and his group's work really catch our eyes. They use the targeting ability of human neural stem cells to deliver IFN-β into glioma sites. But firstly, they overexpressed IFN-beta and cytokine dynamics in human neural stem cells. Then they used these neural stem cells and 5-fluorocytosine to treat animal models and successfully prolong the survival periods. Inspired from their work, we would like to use our culture system to better study how IFN-beta affect human neural stem cells and human glioma stem cells. And different from the cell lines we mentioned before, our culture systems are from primary tissue, which is closer to the in vivo situation. So in our project, we would like to first study the functional role of IFN-beta and then to investigate the differences between these two cell lines. Finally, we hope to identify some potential molecular mechanism. Now I will share our results with you. At the beginning, we used the different dosages of IFN-beta to treat human glioma stem cells. To testify the optimal dosage, we controlled the dose lower than 11 nanograms per microliter at first. From the picture, we noticed that human glioma stem cells typically display two types of cell morphologies, sphere-like status and adherent status. We used the image J to quantify the area size of both status, and we can see here the 11 nanograms per microliter IFN-beta could successfully decrease the sphere-like area and also adherent cells. To compare the differences between neural stem cells and glioma stem cells, we performed ASA again with a higher concentration up to 100 nanograms per microliter IFN-beta. As we can see here, higher dose keep inhibited human glioma stem cells growth and decreased the coverage ratio of adherent cells, but no significant morphological changes were observed in human neural stem cells. This result proved that neural stem cells can maintain stability after IFN-beta treatments and somehow support the clinical usage of human neural stem cells for delivering IFN-beta in glioma treatment that we mentioned before. Then we performed immunohistochemistry to detect the expression of Ki-67, S100-beta, and SOX2 in human glioma stem cells after 6 days of IFN-beta treatment. Here are the 4 times pictures. We also included 10 times photo to better observe the number and status of cells. Similar to our morphological observation, the number of Ki-67 positive glioma stem cells decreased after IFN-beta treatment. At the same time, the total number of cells and the number of S100-beta and SOX2 positive cells decreased as well. Because the total cell number decreased, so the ratio of Ki-67 to dye staining did not show a differences. Overlapping images showed that the relative expression level of S100-beta increased significantly. The increased S100-beta was agreed with the decreased proliferation because S100-beta can stand for as a marker for astrocytes. So from the magnified pictures, we also noticed that the size of cell nuclei increased. The higher magnification image of DIP revealed the nucleus status. We draw a diagram of the nucleus and calculate the area of them. The results revealed that the size of the nucleus increased in a dose-dependent manner following IFN-beta treatment. In clinical, IFN-beta is administered over a long time course, so we designed a continuous treatment to examine the long course effect of it. Both glioma stem cells and neural stem cells were cultured in either control medium or IFN-beta medium for 1 generation. 6 days later, we got 3 groups after passage cells to new medium such as control to control, IFN-beta to control, and IFN-beta to IFN-beta. We take pictures on day 2 and day 8. So from the day 2 data, we can see the number of sphere-like cells decreased in continuous treatment group as comparing to others. Although no significant effects were observed before in human neural stem cells following short-term stimulation, continuous stimulation successfully decreased the cell growth here. On day 8, the cell keeps the trends of day 2. Same as before, we calculated the sizes of both cell body and nucleus. Significant enlargement can be noticed with continuous stimulation. The glioma stem cells in the continuous group displayed an oligodendrocyte-like morphology with a long and massive synapse. Quantitative analysis was performed on the surviving clones, and significant reduction of the coverage rate and single clone area was observed. After the morphological observation, we performed RNA-seq on samples treated with or without IFN-beta in both glioma stem cells and neural stem cells. The analysis of different gene expression was used to identify the genes that were up or downregulated. Here we named the different genes in glioma stem cells as glioma positive and negative genes. Also in the nuclear stem cells, we have nuclear positive and negative gene groups. And then we drew Venn diagram to select the genes only upregulated or downregulated in human glioma stem cells, but no change in human neural stem cells. The GO analysis revealed that the glioma stem cell positive genes were primarily enriched in cytokine-mediated signalings, immune response, response to external stimulus, and cell adhesion pathways. The KEGG analysis revealed that the negative genes were enriched in the cell cycle and ribosome pathways, where overall genomic analysis results agree with the previous morphological observation, supporting the increased cell adherence and the decreased cell growth ability. The expression pattern of the immune response and cell adhesion genes were displayed in heat maps. Consistently with known IFN-beta downstream pathways, JAK2, STAT6, and NF-κB1 and 2 were identified within the immune response. We also explored the detailed expression patterns of 22 genes in the cell cycle pathways. Typical cell cycle-related genes such as cyclin A2, B1, B2, and D1 were significantly downregulated following IFN-beta stimulation in glioma stem cells, but remain unchanged in neural stem cells. Here is the graphic abstract of our data as we summarize all the changes in morphological ways and genetic ways. Again. Well, in our study, for each cell type, we only include 2 cell lines for RNA-seq. So further genomic analysis with multiple cell lines or primary tissues from these 2 different cell types may help us better select the keyword candidate genes as potential therapeutic targets and better understand the molecular mechanism. At the same time, we already designed and performed experiments to test the different dosages of IFN-beta stimulation combined with regular chemotherapy drugs such as TMZ in glioma stem cells. This result can be more helpful for clinical consideration. At the end of today's talk, I would like to send my deeply acknowledgement to Professor Gao, Dr. Han, and all the members from Gao's lab. Well, that's all about our sharing today. Thanks again to the kind invitation from Cell Regeneration. Any further discussion about our work is welcome. Please feel free to contact us from email. Thank you.